Hello, my name is John Moyer, and I want to welcome you to the NIH Office of Disease Prevention's Methods Mind the Gap webinar series. This series explores research design, measurement, intervention, data analysis, and other methods of interest to prevention science. Our goal is to engage the prevention research community in thought-provoking discussions, to promote the use of the best available methods, and to support the development of better methods. Before we begin, I have some housekeeping items. You can submit questions during the webinar via the Q&A pod. You'll need to open the pod by hovering your cursor at the top of the Teams window and then clicking on the Q&A icon. We will open the floor to questions at the end of the presentation. The slides and video recording will be posted on our website, prevention.nih.gov/mindthegap, in a few weeks. At this time, I'd like to introduce today's speakers, Dr. Nori Hyun and Dr. Brian Williamson. Dr. Nori Hyun is an Associate Biostatistics Investigator in the Biostatistics Division at Kaiser Permanente Washington Health Research Institute. Her research is focused on methodological development and survival analysis, nonlinear modeling, complex probability sampling, and measurement error, with applications to cancer screening and risk prediction using electronic health records. Since joining KPWHRI, Dr. Hyun has expanded her work to include pragmatic clinical trials and research on substance use disorders. Dr. Brian Williamson is an Assistant Biostatistics Investigator at KPWHRI. He conducts statistical methods research on using machine learning to make more accurate and efficient use of data. He also collaborates on drug and vaccine safety and effectiveness research, pragmatic clinical trials, and mental health research. It is my pleasure to welcome Dr. Hyun and Dr. Williamson.
All right, we'll get started. Thank you, Jonathan, for the kind introduction, and thank you, everybody, for being here. I'm going to start off. So the first part of our presentation is talking about design considerations for subgroup analyses and cluster randomized trials, and then I'll hand off to Nori, who's going to talk about analysis considerations. First of all, I want to acknowledge my wonderful collaborators who I did this work with, Yates Coley, Clarissa Sue, and Andrea Cook, all at Kaiser Permanente Washington Health Research Institute, and Courtney McCracken at Kaiser Permanente, Georgia. And I also want to recognize our funder, the Patient-Centered Outcomes Research Institute. So to set the stage for today, wanted to start out by talking about very broadly what are heterogeneity of treatment effects. This may be review for some of you, but I wanted to sort of start us out on a baseline. And I'll use the abbreviation HTE to shorten that phrase. So what is HTE? It's a differential effect of an exposure or an intervention across subgroups. And I'm using the terminology heterogeneity of treatment effects deliberately because I don't want to necessarily imply a causal or a biological interpretation if HTE is present. There can be other mechanisms through which HTE can be present in a given population. And if HTE is present for your exposure or intervention effect, you can then prioritize resources to try and either maximize benefit or reduce risks, or both. And you can also try and determine whether your intervention remediates or exacerbates health disparities. And of course, we're all interested in trying to do the best possible medicine in public health, and so trying to make sure that we sort of direct resources in the right way and make sure that our interventions are tailored such that they are providing benefit to the most patients is very, very important, which is why, of course, HTE is important to characterize. So as a case study throughout my portion of the presentation, I'll be focusing on a trial called INSPIRE, which was designed to improve COVID-19 vaccination rates at long-term care facilities. So as a little bit of background, rates of COVID-19 disease and death were higher in long-term care than in the general population for much of the pandemic. In fact, the first sort of large outbreak of COVID-19 in the US was in Kirkland, Washington, which is in a long-term care center in Kirkland, which is just a little ways away from where I am today. So this is a high-risk population. These are typically older adults that are living in long-term care. And so we really wanted to do a good job of protecting them. And sort of going along with this, there was a lag in COVID-19 vaccinations among long-term care workers, the staff members who are caring for folks in long-term care and working at the long-term care centers. And, you know, one thing to keep in mind, sort of as context, is that long-term care workers are often low-wage workers, and they represent a very diverse social, cultural, racial, and ethnic background. And so making sure that any interventions that we were designing to try and get people to increase their vaccination rates represented that and were sort of tailored in some way. So very briefly, the study design, we enrolled 40 long-term care centers, approximately 4,000 staff members. We recruited from 2 states in the US, Georgia and Washington State. And we tried to recruit sort of both, geographic diversity, but also within those states, a little bit more diversity, so focusing on urban areas like Atlanta or Seattle, and more suburban or rural locations to try and get a mix of people who were coming from many different backgrounds. The primary outcomes of INSPIRE, the first primary outcome was the COVID-19 booster rate. This is a cluster level outcome, right? So we have each of the long-term care centers and we get 1 booster rate from each of those centers. We chose this primary outcome primarily due to concerns about data capture. So we were thinking that people might respond to a survey that we sent out to all eligible staff members who worked at any of these centers. But they might not always disclose their vaccination status, and they also might not respond. But on the other hand, we knew that these centers were tracking booster vaccination rates for people who worked at the centers in a more anonymous way because they were required to report these booster vaccination rates to the Center for Medicaid and Medicare Services. So we knew that we would have good data capture on this cluster level outcome, and this is of course something that we cared about quite a lot because we wanted to increase booster vaccination. And I'm saying booster because the original study design was to try and increase the primary series vaccination rate, but there was a vaccine mandate instituted in Washington State for long-term care staff sort of right before we were about to start the study, and so we changed our outcome to the boosters, which is a little bit extra context for results that I won't be talking about today, but you can check out for this trial. The second primary outcome is the long-term care center staff net promoter score, which is an individual level outcome. This is measured using a survey, and this net promoter score is sort of comes from market research, and it's a Likert scale, a 10-item Likert scale, where you're asking people, what's your willingness on this scale of 1 to 10, 10 being the best, to go out and promote the booster vaccine. Tell other people that they should get it. And the particular one that we were interested in was how likely are you to go out and tell your coworkers that they should get vaccinated, with the idea that if you were willing to promote, then hopefully you would be increasing the booster rate at your center. We compared 2 interventions. These were both booster promotion materials. On the left-hand side, I'm showing usual care, which is standard US government materials. So here is an example of a poster from CDC. And on the right-hand side was our sort of intervention, what we called co-design intervention, where teams of staff worked together to design, along with people from our study, to design booster promotion materials. And so here on the right-hand side are some of the examples of posters that were put up around long-term care centers that were designed by staff to try and get other staff members to increase to go out and get their booster. A little bit more about co-design. This is about tailoring messages to specific audiences. And in INSPIRE, we focused on language, cultural, and ethnic affinity groups because we thought that these would be quite diverse, and also, right, that people, through their lived experience might be coming at the question of whether or not to get a COVID booster in different ways. So we measured race and ethnicity, allowing for 57 total options for people to choose between, which was quite granular. But this was really important for both creating our co-design groups and also then the end goal of trying to have messages that were tailored to different groups of people. And so especially in this case, but also, of course, in many other types of interventions, it's natural to think about doing an HTE analysis. And so in Inspire, we planned to do analyses based on which state you were in, sort of the urbanicity of your location, and whether or not the baseline booster rate at the site was above or below the median in that state. All of these are cluster level outcomes, and so it sort of made sense to look at this. We were also interested in looking at self-reported race and ethnicity, but especially because we were tailoring our messages to folks from different backgrounds. But the first and main problem with this is that neither the sites nor any other entity had comprehensive and accurate race and ethnicity data for staff. And we were really worried that we wouldn't get necessarily a representative sample from the staff survey of people from different groups. So that was our primary worry. And also, we didn't know at the time what it would look like to try and do an HTE analysis of this individual level variable, which is your self-reported race or ethnicity, and this cluster level outcome, which is the COVID-19 booster rate at the center. And so that's what I'll be talking about today, is this sort of tension between this individual level covariate and this cluster level outcome. Okay, so thinking a little bit first just at the individual level, we have data on N sub C staff members in each cluster. C ranges from 1 through K. And this is I'm setting up sort of how we would do an HTE analysis in this individual level data set, and then we'll move to the cluster level data set. So A is an indicator of intervention assignment. That's the same for everybody in a cluster. Z, I'm calling a vector of cluster level subgroup variables. So these are things like your state, whether or not your cluster is in an urban or a rural location in Inspire. W is baseline covariates that can be individual level. So in this case, this could be your self-reported race or ethnicity. And Y is a binary outcome. So in this case, it's whether or not you got your COVID booster. If we're interested in assessing HTE by a single binary variable, W, let's say that our target estimand is the difference in risk difference based on levels of W. So we could say, okay, we think in the population there's a linear regression model relating our outcome, our binary outcome Y, to the intervention and the covariates. And we have this linear predictor on the right-hand side. And so under some standard assumptions, the regression coefficient beta 4 quantifies the HTE. It's this difference in risk difference based on levels of W. And we can estimate this and we can make inference on this sort of in the standard way. Okay, so now moving to the cluster level, right? So now we have to sort of redefine things and roll things up from the individual to the cluster level. So now I'm defining Y bar as the cluster level outcome proportion. So this is the COVID-19 booster rate in Inspire. A, again, is the indicator of intervention assignment, which doesn't change, right, for individuals within a cluster, so this rolls up easily. Z, again, are these cluster level subgroup variables like state, like urban versus rural location, so those also roll up very easily to the cluster level. And I'm going to call X baseline covariates that we may have had to do some aggregation for. So these are things like, in the Inspire example, self-reported race or ethnicity, though we didn't do that in Inspire, but you could think about it that way. So there are many different ways of aggregating this individual level covariate that I'm going to again call W. These are just two possible ways that I'm showing here, but you could think of many more. So one is to take the overall mean-centered proportion. So X is equal to V minus the grand mean across all clusters of the V's, where each V is the proportion at that individual center. Another way that you could do this, so this right, so in the race and ethnicity example, this would be the proportion of folks who identify as one particular race or ethnicity. Another way to do this would be to say, okay, I'm going to take that proportion and see whether or not it's greater than some threshold. So you could now be saying for each cluster, is this proportion high or low based on the threshold T that you've chosen. But there are a lot of complications with this type of aggregated data. We've known for quite a long time that individual level effects may not be identifiable if you only have this aggregate data. This is often called ecological bias. And since the late 80s, we've also known that this type of aggregate data can bias the association between your exposure and outcome if you either have confounders that have been aggregated to the cluster level, or if you have HTE variables that have been aggregated to the cluster level. And that's for this exposure outcome association. We also know that if you have compositional data, which arise when you have mutually exclusive binary variables, so like in the case of race and ethnicity, we often encode that as binary variables that are mutually exclusive, saying which race or ethnicity category do you fall into. So now if you come up with proportions, you have compositional data where if you change the value of one proportion, you're necessarily changing the value of at least one other proportion, and because the composition is constrained to lie within 0 and 1. And so this can make interpretation of regression coefficients really challenging. Okay, so now thinking about HTE with these cluster level outcomes. So our outcome y bar is this proportion. We're interested in HTE by a single variable x. Maybe it's been aggregated. Our target estimand, again, is the difference in difference of proportions now based on values of x. We could, again, think about this population linear regression model where y bar is related to, which is the cluster level outcome, is related to our exposure and these other covariates. And we could make some assumptions again and say that alpha 4 quantifies now the HTE. But the way that we've done the aggregation here determines how we interpret this alpha 4. Right, so it depends on if we're using the mean centered proportion or the threshold. If W is compositional, then we have to factor in how we've transformed the categories to enable model fitting. Oftentimes you have to drop one of the categories, but that sort of makes things much more complex. And then thinking about what a 1 unit change implies is really, really difficult. And so this, not only is fitting this model more complex, but interpreting this model is more complex. Okay, so now I'm going to show some simulation results that sort of get at some of the problems with trying to, in particular, if you were thinking about designing a study and trying to do a power calculation to see, what's my power for HTE, for example, which of course oftentimes we're not powered to detect HTE, but you may be interested in looking at that anyways. Some of the pitfalls that can arise from considering your effect on the cluster level rather than considering it on the individual level and then aggregating to the cluster. So here I'm generating data that mimic the INSPIRE trial. We sample a number of clusters. We designate them as urban or suburban or rural. We do constrained randomization of these clusters into intervention arms. Each center has staff members that each have whether or not they received their booster vaccine and also have their self-reported race. Here I recorded that just as a single binary variable to make things a little bit easier to look at, but it would get much more complicated and difficult if you allowed multiple categories. So then once we have that individual level data, we roll up to the cluster, taking the average of the booster rates for our outcome, and then defining this center level proportion and an indicator of the proportion of race and ethnicity greater than 1/2 as the two ways we're going to roll up race and ethnicity. Because we have both individual level and cluster level data here, we can fit regression models at both levels, and we can look at power and type 1 error for testing for HTE by self-reported race to see what happens when you are thinking about running each of these models. Okay, so here is the first slide of results. I'm going to spend a little bit of time here because it'll set up the rest of the results. So here I'm showing type 1 error for testing for HTE by self-reported race, which again is a binary variable, a single binary variable here that I'm aggregating to the cluster level. On the y-axis, I'm showing the empirical rejection proportion, which for a 0.05 level test should be about 0.05. So this is the average over 1,000 simulations. On the x-axis, I'm showing different regression models that I'll talk a bit about in a moment. And then the panels, I'm showing different scenarios, which are encoding whether or not there's true differences in booster rates or true HTE. And in this case, I'm looking at type 1 error, so there's no true HTE by race or ethnicity in any of these scenarios. The models are encoding whether or not I'm running an individual level model, so that's things that have a 1 next to them, or a cluster level model, which is the things that have a 2 next to them. And then the letters are denoting whether or not I'm adjusting for self-reported race and allowing for HTE by self-reported race and also by urban or rural status. So we included those sort of as a negative control because that's a cluster level variable. So we should have pretty good results both for type 1 error and power for that cluster level variable. That is sort of truly defined at the cluster level. So what we're seeing here is that for the individual level models, so that again, those have the 1 next to them, type 1 error is controlled across all of these scenarios. So that's good. We had the individual level data, we have proper type 1 error control. For some of the cluster level models where we've aggregated this race or ethnicity variable, we're seeing a little bit of inflated type 1 error. Sometimes it's controlled, sometimes not, depending on the scenario. And always the cases where we're seeing inflated type 1 error are the cases where we're using the proportion, the center level proportion of this race and ethnicity rather than the threshold value. Okay, now moving on to power. So these are scenarios, again, where there's true HTE by self-reported race. And in this case, the value of that regression parameter, so we're generating data under these true regression models. The value of that regression parameter is quite large, such that we would hope, at least in the individual level model, to have some power to detect that HTE. And we see here, so again, these are the same models. 1 is individual level models and 2 is cluster level models, and then the same ways of adjusting for, in the cluster level, accounting for aggregating race and ethnicity. So I'll just go to what the models are here. And so we're seeing that in these scenarios, when there is true HTE by self-reported race, we have really high power to detect that in the individual level models, and we have uniformly low power to detect that in the cluster level models. So all of these, even though there is true HTE at the individual level, it doesn't show up at the cluster level. So what does this sort of imply? And I'll close out my section with some thoughts about this. So, right, if you have individual level data, this is the best way to proceed. You should do this whenever you can because you have high power to detect large HTE effects and you have controlled type 1 error. So of course, this is the best thing that you can do. In cluster level analyses, we have even lower power to detect HTE by an aggregated individual level variable than we would if it was a cluster level variable, which of course we always have lower power than for our intervention effect. And we also are seeing elevated type 1 error when using the proportion, when we've aggregated using the proportion in some settings. And so one of the key things that I want to highlight is that if you're designing a study where you have a cluster level outcome and an individual level covariate that you're interested in HTE for, you wouldn't have known that you had such low power or possibly elevated type 1 error if you simulated data at the cluster level, right? If you said, this is my regression, my possible true regression model, these are the effects that I think will happen, and so I'm going to generate data directly from that, you wouldn't sort of see that, right, this true HTE, which occurs on the individual level, will sort of go away in some sense in the aggregate, and that you have incredibly low power to detect that effect. But one other thing that I want to mention is oftentimes, like I mentioned, we know that we don't have large power to detect HTE, but we can still, if we see meaningful differences, whether that's clinically meaningful or meaningful from a public health perspective, we can still act on large estimated differences, even if that the test for HTE is not statistically significant. And at the bottom, I have a QR code with a link to the paper, which you can check out if you're interested. And now I will turn it over to Nori for the second half.
Thank you, Brian. And I'm very happy to make a presentation partnering with Brian. And then in part 2 of our talk, I will talk about type 1 error inflation and control for detecting HTE in cluster randomized trials when using mixed effects models. Please, next page. So this work is joint work with my collaborators, Jennifer Bob, Abisola Idu, and Andrea Cook. 4 of us are all KPWHLI, Kaiser Permanente Washington Health Research Institute. And then this work was funded by NIDA, National Institute on Drug Abuse. Next slide, please. Okay, so I will start by briefly introducing the motivation of the case study, which comes from primary care opioid use disorder treatment called the PROUD trial. PROUD trial is a pragmatic and two-arm with a 1-to-1 ratio cluster randomized trials, and the trial implementing the Massachusetts model of nurse care management for opioid use disorder with the goal of increasing access to OUD treatment in diverse primary care settings across the United States. So in the trial, 6 health systems participate in the trial, and then within each system, 2 comparable clinics were recruited and randomly assigned to either the PROUD intervention arm or the usual care arm. So in total, 12 clinics were included and serving as a cluster unit at the clinic. And the cluster size, that means number of patients per clinic, varies across clinics. So for example, median number of patients per clinic was 122, and then with the interquartile range from 97 to 220 at baseline. And then this, the primary paper was published in JAMA Internal Medicine in 2013. So if you are interested in more details, then you can find it from the paper. And the PROUD trial has one primary outcome and have several secondary outcomes. And one of the secondary outcome was the number of days patients used acute care services, which is count data. And then I would like to share a very interesting experience we had in analyzing the secondary outcome. So next slide, please. As a part of subgroup analysis, we plan to fit a generalized linear mixed effect model to detect the heterogeneous treatment effect by sex. So here for subject J in cluster I, Y subscript IJ, that is represents number of days of utilization of acute care services. And using logolink, we fit a GRMM. So for the GRMM in the model for fixed effects, we included a baseline value and cluster level intervention effect, here's treatment, and individual level sex indicator, sex difference. And for the interaction term, we included the interaction between treatment and sex to capture the HTE. And we included the overall random intercept due to clinic, but did not include any random intercept due to system, because within system we have only two clinics, so repeated measures is very limited. So yeah, would you go to the next, Brian? Yes, here. So this table presents the results from the fitted GRMM, and the main primary intervention effect was not significant, but we did find a significant sex interaction effect here. The p-value is 0.01. And we also calculated this intervention effect within men and within women. So for men, the intervention effect is 0.07, but for women is 0.21. But in both cases, confidence intervals included a 0, suggesting that the intervention effect is null. But what puzzled us was the p-value. The intervention effect was 0.01, very significant. However, the confidence intervals of the treatment effect within men and women substantially overlapped. So that's like a question mark why that is so distinctive results. And then one hypothesis was probably the p-value would be type 1 error. And then because we use the Wilder test statistic, and then another possible explanation is a standard error estimate for sex interaction would be underestimated. Or I mean, also we applied some small sample size correction methods. So that is also a question mark that is well controlled. So we wanted to test this idea through a simulation, but before doing so, we also conducted a literature review. And then next slide, please. So we found a similar issue type of error inflation for detecting HTE in not clustered using mixed effects model that has been reported and studied by Baer and colleagues in 2013. But there are some their settings are quite different from cluster randomized trials because their field is more focuses on psychology, science, and then so their cluster is person level, not clinic, and also they include several intervention effects within the same person. So there was, and also they assumed moderate to large number of clusters. So there were research gaps in the standpoint of small number of clusters, and also they only focus on continuous outcomes. So they did not study any non-continuous cases. So we wanted to investigate this question and then fill the research gaps. So we conducted a simulation study. Next slide, please. In the simulation study, we considered three different outcome types, including continuous, count, and binary data. And then for regression models, we compared three. One is a marginal model, GEE, generalized estimating equation with the independent working correlation. And the other two are conditional regression models. One is GRMM, and the other is GRMM2. So more specifically for GRMM, we included only overall random intercept due to the cluster, only one intercept. So that can be called within clinical correlation. In GRMM2, we included two random intercepts. One is two random effects. One is a random intercept due to the cluster, and then random slope for subgroup. But here is a subgroup. We just consider binary or dummy variables. Then that can be translated into cluster by subgroup correlation, or subgroup specific correlation was specified in GRMM2. And also, compared to GRMM2, for example, binary case, we add two more parameters because we are modeling the bivariate normal distribution assumption for two random effects. And then we also simplify the simulation or data generation setting. So we assume very well-balanced cluster randomized trials. Actually, in practice, it's not true, but I want to reduce some more complexity. So we just only account for intervention, cluster level intervention effect, and individual level sex binary subgroup factor here. Go next slide, please. So we considered a total 7 simulation settings, and yeah, we just considered very thorough, diverse settings. So particularly scenario 1 through 3, the true data generation model is GRMM2, and then we wanted to test how much that is worse when ignoring cluster by subgroup correlation when fitting linear mixed effect. Generalized linear mixed effects model. And for three different outcomes, we borrowed the information from the Proud study data. So for continuous and count and binary covariance and all fixed effects are different setting. And also we considered the moderate size 50 of number of clusters, large number of clusters 100. And also Proud trial cases, we also considered number of 12 number of clusters, and then with the fixed size of cluster size or varying cluster size, we also considered in the simulation. Yes, I will give you more details when we just digesting the simulation results. But also we the other way, if the true model is GRMM and then fit GRMM2, that means GRMM2 is over fitted the cases, then how much that is losing power, and then how control the type of error rates. So would you go to the next slide? So yeah, I will skip it, and then because I can give more details each table. Next slide, please. So this is a simulation 1, the results. So that means this is a true data generation process is GRMM2. So that means there are truly cluster by subgroup specific correlation within cluster, and a very ideal setting for GRMM2. That means moderate to large number of clusters, and also the random effects is quite size, so adequate. So in that case, we compared the three models, GEE, GRMM, and GRMM2. So GRMM, and then also two things, color. Blue color indicates well control at the nominal level of 5% within range of 4 to 6%. 4 to 6%, and then red color indicates type 1 error inflation beyond or above 6%. So we can, across all three outcomes and even 50 and 100 and above clusters, GRMM exhibited type 1 error inflation up to 47%. But GRMM2 and GEE controlled well the type 1 error rate at the nominal level, particularly when sample size or number of clusters is 100. However, it's a little bit surprising to me. GE exhibited a little bit higher type 1 error inflation when sample size number of clusters 50. So that is a well-known fact. When sample size or number of a cluster is small, GE exhibited a little bit type 1 error inflation. However, number of cluster 50 is generally not considered as a small number of clusters. The next slide, please. And this is the result from scenario 2. Similar, but only one deviation from the scenario 1 is we considered the very small random effects. The other are the same setting, so we reduced the downsize the random effects by 10%. So because the correlation, the random effects is very small, GRMM just decreases the type 1 error inflation compared to scenario 1. And board fonts indicates conservative type 1 error. And then in the bracket, the number indicates the total number of singular fit issues we encountered through 1,000 simulation results. Because we reduced the random effect sizes, so that increases the singular fit issues, but we cannot completely avoid it when even fit GRMM with a simpler model. And then we can see at 100 case number of clusters, GRMM2 showed very conservative control for type 1 error rate, and there was a singular fit issues. So we, in this case, we also consider two-step procedures. First is fit GRMM2, and if we encounter singular fit, then fit GRMM. So the two-step procedure type 1 error rate is a sort of weighted average between GRMM2 type 1 error and GRMM, because we just switch it after encountering singular fit issue. So G showed well control, and the two-step procedure also showed well control in this case. Next slide, please. For scenario 3, we just changed allocated random effects, but reduced the number of clusters to 12 with a fixed cluster size or varying cluster size. So mostly GRMM2 and then two-step procedure. I think it's GRMM2 showed well control overall in this case. But even GEE, because it's a small number of clusters, that's the reason GE does not inflated some type 1 error rate. Next slide, please. This case, so far the true data generation model was GRMM2, and then this time true model is GRMM, and then GRMM2 is overfitted case. In this case, so GRMM2 is overfitted and then controlled very conservatively type 1 error when sample size is 12 and then with the varying size. However, that works well, but relatively conservative when number of clusters 50 with a fixed size. And GEE is the same issues. I don't want to say it repeatedly. Okay, the next slide, please. And this is the result from scenario 5. Because what if the true model is GRMM and then still GRMM2 is overfitted, but if the subgroup level is not binary, this case is 3, then how it would be like maintain the same about the type one error control. And then the GRMM2 showed very conservative type one error control when varying cluster size. But that is works well. But in this case, GRMM, because GRMM is the true model, and then that is like a reference control well. But the two-step procedure works better than GRMM2. And next slide, please. So this is the final results of the scenario. This case is GRMM2 also misspecified. That means we consider the true data generation models having heterogeneous correlation structure by arm. So GRMM2 as well as GRMM both are misspecified. In this case, GEE showed even like a GE and the GRMM showed exhibited type one error information, most cases, and then but when number of clusters 12 with the varying size, GRMM2 showed conservatively or well controlled type one error rate. So the next slide, please. I didn't say specific what small sample size correction methods we use, particularly when considering number of clusters 12 with a fixed or varying number of clusters. So we considered the 4 options for mixed effects model cases, and Seroth-White correction method within denominator degrees of freedom and subtracting the number of cluster level fixed effect from the total number of clusters. And the last one is the parametric bootstrap method. So we compared these 4 small sample size correction methods and then chose the best one for each scenario. And for GE, we used Fay-Graubard small sample size correction methods. So for both, we used 2 R packages. One is SA-W source, and the other is parameters for R package. So for continuous outcome, regardless of what is true model, what is a fitted model, consistently Seroth-White small sample size correction methods outperformed. But for count and binary case, the best performer differed depending on the situation, what is a true model, what is a fitted model. So I will summarize in the last page. Okay, next slide, please. So this is the results, the comparison of 3 models and applied to proud trial data. And the results in the center is exactly the same I showed at the beginning from the GRMM. And the left side is from GRMM2 and right side from GEE. So the interaction effect or HTE is, differed. So GRMM, that is like a significant interaction effect, but GRMM2 and GE showed a non-significant effect. But one another thing interesting I found was the intervention effect direction was changed by the models, even though those are indicating null effect. However, for example, like a GE within men and then within women, the intervention effects is like negative 0.18 and then negative 0.2. But the GRMM, that is like a positive, so that is totally the opposite. Okay, the next slide. So I want to summarize all the findings from the simulation into this chart. So in this flow chart, 2 questions we can ask. First diamond diagram, whether there are truly exist cluster by subgroup specific correlation, and if there is present, then the random effects or the size is adequate. That is number 1 question. And number 2 is number of clusters is greater than 50 or equal 50 or fewer. There are 2 questions we need to ask. So in overall, if we have, because the number 1 diagram questions we don't know in practice, we don't know what is a true model, but we just the number of clusters we know. In that case, if we have less than 50 or equal to 50 number of clusters, overall two-step procedure is preferred and recommended. And then if we have greater than 50, then GE or two-step procedures are preferred. So yeah, so that is the end of my slide, 45 minutes. Yeah, thank you. I'm done.
Great. Well, thank you so much, Brian and Nori, for that amazing presentation. Got a few questions to ask. So I guess maybe this is more for Brian as it pertains to the first part. But so what would the main pitfall be if you ran the power calculations for simulated cluster level data when you're truly aggregating individual level variables?
Yeah, that's a great question. So, oftentimes when we're designing power calculation simulations, or trying to do power calculations, we say, okay, what's sort of the hypothesized effect of the variable that I care about on my outcome? And in this case, it would be what's the sort of hypothesized HTE, right? I want to say I have power to detect HTE of this level with this sample size. And so if we were going to do that and we started and we said, okay, so my data generating mechanism for the simulations is going to have the cluster level variable, and then there's going to be some coefficient on that, right? We would say, and we would design our simulation and we would run everything and we would say, okay, yes, I have power to detect an effect that's this large. But sort of what my simulations were showing and sort of what I think, oftentimes when we think about designing simulations of any kind, we sort of start from first principles of what the data, like how did the data come to us, right? And in this case, if we are aggregating an individual level variable, the data come to us, well, the data are coming to us as aggregated, but the data actually exist at the individual level. And so in that respect, the real effect or the real HTE is on this individual level association. And so that's where the trouble comes in, because if you hypothesize your effect at the cluster level, you can run sort of a standard power analysis and you'll get an estimate of power for that. But that's not sort of, in my view, that's not like the real power, because the real power is for an effect on the individual level that then you're aggregating to this cluster level just because that's the data that you happen to have. So hopefully that starts to answer the question.
Yes, thank you. Question maybe for part two for Nori. So we have this two-step procedure that you recommend when you have a random effect for subgroup slope that's like small or null. Is there like a formal test that one could do to assess whether there is a random slope by subgroup? Or, go ahead. Sorry.
Yeah, that's a great question. We've thought about that. What would be the best approach to more formal testing? But tricky part is power, because even though they are truly exist, but not significant, then should we skip it? So that's the reason we just consider two-step procedure. If we encounter any singular fit issues or small random effects, then switching to GLMM rather than doing exact test, because generally we just all the time lack of power for HTE. So that's our thoughts. Thank you.
Great. So this study was kind of a parallel group cluster. The examples you did in the simulations were a parallel group cluster randomized trial. Do you think similar issues or similar solutions would be present for like designs like step wedge designs?
Yes, definitely. I think because it means step wedge is a sort of some more extension of parallel cluster randomized trials. So the tricky part is what I found is in cluster randomized trials, other patient level characteristics is hardly to make some balance. And then that is like a tricky part. And also even within cluster, there are possibly more complex structures. So I think that can be also happen in cluster, step-based design.
Interesting. They're definitely much more complicated correlation structures
Yes.
and variance structures in those designs. Yeah, definitely. So some of the simulations had like a large number of clusters, like 80, I think was one in Brian's. I think there was like 100, maybe there is in yours, Nori. It's like in cases where you have like a much greater number of clusters, do you think that you could get power for cluster level analyses if you have like tons and tons of clusters?
Brian, do you
Yes,
want to?
I'll jump in first. I think that's probably right, but I guess, so in our simulations, 40 was the number of clusters that we had actually in Inspire, and it was like nearly impossible to get larger than that. We tried our hardest and couldn't get any more, and so 80 for our simulations was like, wow, this would be amazing. What if we could get 80 clusters? And so a lot of times, it's just impossible to do that. But in cases where you are very lucky and somehow have tons of clusters, I think, yeah, you could be seeing power in those scenarios. I'm hesitating a little bit because there still would be this issue of, right, what happens in my example, what happens when you're aggregating this individual level variable. So I don't know, like, it would probably depend on the scenario, or like what the true HTE actually is, how that would sort of interplay with the number of clusters. But it does seem to me like there should be some number, maybe it's really, really large, where you still have power.
And also I want to add one more comment. So covariate adjustment techniques for improving power and for main effect, that is well studied, but that is limited for HTE analysis. So instead of, if we cannot fully, increase beyond I think even 40 is like a very, very happy case, lucky case. But yeah, so another way we can consider some including covariate adjustment strategies or techniques to improve the power.
Great, thank you. In terms of the correlation structure used, what is the rationale for using independent working correlation in GEE as other than like unstructured or AR1, other types of
the rationale is because we don't want to more dive into the improving the power for GEE. So that's the reason, like there's comparing like an independent working correlation versus a more structured or specified working correlation differences like regarding the power, but our target or more focus is on type one error and null effect. So that's the reasons we did not go further about GE specific working correlation specification. But in flipping side, if we have interest, while controlling the type one error rate at the same time improving the power, then definitely we should consider more specific and more correctly specific correlation structure for GEE.
Great, thank you. One of the like the subgroups that you analyzed were basically dichotomous. I think like either of a certain race or ethnicity or not. But of course, there could be many more. I think in the when the original paper for the special issue, I believe there were like 57 different categories. So that's what would I guess how generalizable do you think the findings are to cases where you have multi-level subgroups?
For me, yeah. So from my end, my suggestion, because key thing is GRMM2 is like how much we want to spend a number of parameters. For example, like a multiple case, even level 4, then we cannot assume any simpler compound symmetric covariance structure, particularly for race and ethnicity. That means the upper triangle number of parameters we have to estimate it. So my suggestion for random, if we just need to include random slope for like a race and ethnicity, need to thinking about some collapsing. So that means because generally continuous variable that is weird, but the binary or dummy variables we can collapsing for random slope. So my suggestion is just collapsing the race and ethnicity or just reduce the level for categorical cases.
Yeah, and in my view, I think it's yes, I would add that it's both about sort of an increasing number of parameters that you're trying to estimate, which will probably necessarily reduce the type 1 error, especially if you're looking at, HTE, by multiple levels. But I think it will. So not only are you increasing the number of parameters, but you're also increasing sort of the difficulty of interpretation in some respect, and also I would expect that what we observed when you were using the proportion, which we saw in inflated type 1 error rates and very low power, I would expect that might get worse. That would be my hunch if you have more categories, because of this problem where changing the level of in this linear regression model, changing the value of one necessarily changes the value of others, and so I think that it seems like it would get more difficult to estimate and more difficult to control either type 1 error or have nice power, but that's just a guess.
Thank you. So for the GEE, the small sample correction approach was the Fay and Graubard. I know of some other approaches. Cowerman and Carroll is an adjustment that comes to mind. Do you think there could be any difference in results based on the type of small sample correction employed?
Yeah, good question. But I just did not include the other cases. But definitely, we need to do more study. But I found recently published in 2025. I cannot correctly remember where. But yeah, but that showed some similar results, like a type 1 error inflation. So that is an interesting topic still for what is a universally applied to any outcome, not only continuous. I mean, continuous, I think that is okay, but binary account or non-continuous outcome, and particularly for small sample of clusters, that is still like an open question.
Great, thank you. And I think we have time for one more question. So for a two-level model using two-level modeling using a mixed model, if there's a random component in the mixed model defining cross-clinic variance, is it also needed to consider the variance covariance structure among subjects within the same clinic?
So that means like a heterogeneous by arm within cluster, or so by arm and by subgroup?
I'm looking at the question now. If maybe perhaps that the speaker or the listener wants to clarify, but it's a random component in the mixed model defining cross-clinic variance.
Cross-clinic.
Do I also need to consider the variance covariance structure among subjects within the same clinic?
Among subjects. Yeah, I mean, good question, but I cannot answer because I don't clearly understand the point of cross-clinic and the variation cross-clinic and then random subjects.
Yeah, usually I consider the clinic. If I consider clinic clusters, and I don't, usually consider cross-clinic. But
do you mean like a system, like like a system and then two levels?
Yeah, like in your case with the two systems.
Yeah. That is a good question. If we have multiple clinics within health system, would it be like an idea? But within health system, only 2 clinics. So that is the reason that we dropped the idea. But if we have multiple clinics within the health system, then definitely, that is a more complex situation though. Yeah, then definitely we need to should consider the covariance structure.
Great, thank you very much. As we're near the top of the hour, I think we'll close things up. Thank you, Dr. Hyun and Dr. Williamson, and thank you to everyone who participated in today's webinar. On the Mind the Gap website, prevention.nih.gov/mindthegap, You will find several resources for this talk, including the slides and the list of references. We will also be posting a recording of today's webinar on our website in a few weeks. Thank you and have a great rest of your day.